Picture this. It's 2 a.m. in a Cambridge lab. A grad student is hunched over the bench, blue desk lamp humming overhead. Standard drug tweak, nothing fancy. Then, a beam of stray light hits the flask the wrong way. The reaction veers off script, and boom, a brand new method for editing medicines is born. Wild, right? Today we're going to unpack that mistake, see why it matters, and figure out how you could steal the playbook for your own molecules. First, the happy accident. The team expected the usual coupling. Instead, the light coaxed two pieces together in a way textbooks said was impossible inside a crowded drug scaffold. Once the shock wore off, they asked the obvious question: could this survive real-world chaos, not just a squeaky clean model system? Spoiler: yes. Why drug molecules are such divas. Medicinal compounds are like overstuffed neighborhoods. Touch the wrong house and the whole block complains. Traditionally, that means weeks, sometimes months of redesign just to swap one carbon. But this, light-guided coupling, it slips down those streets almost unnoticed, edits one address, leaves the fussy neighbors alone. Early runs hinted at three big payoffs: faster backup drugs when resistance shows up, gentler ways to extend half-life, and greener manufacturing at scale. Stress testing the discovery. Here's the thing. Lots of new reactions collapse when you leave the clean lab bubble. So the Cambridge crew did the opposite. They dumped in crushed tablets, sticky syrups, weird formulation gunk, no fancy purification, and they still got crisp new products. That resilience matters. Industrial chemists rarely see pristine samples. They get slurries, fillers, and brutal deadlines. The light trick kept working anyway. Pharma partners are now asking, can this patch stalled projects we shelved years ago? On paper versus in practice. If you look at the mechanism on a slide, it feels almost too simple. Yet in practice, it bends the old rules. Instead of building a molecule from scratch, you take something already packed with rings, heteroatoms, and fragile do-not-touch zones, and drop in a fresh carbon fragment exactly where you want it. The shocker wasn't that the drug survived. It was how many different add-ons the reaction tolerated without grumbling. Moving from showpieces to the real mess. Next signpost, large-scale stress test. The group pulled entire panels of marketed medicines and late-stage candidates, not because those drugs were broken, but because they represent the true molecular clutter industry cares about. Changing the alkyl partner was now as easy as swapping a stock solution. Lengthen a chain here, add a branch there, slide a polar handle closer to a binding site. Medicinal chemists call this mapping the SAR curve. Suddenly, that curve could be traced in days, not weeks. Messy glassware? No problem. Another bonus: the setup is forgiving outside pristine glassware. They ran edits in the presence of binders, colorants, common excipients, exactly the stuff you'd see on a manufacturing floor. Imagine scraping 1 milligram from a pilot batch, tweaking it under room light, then shipping it straight to bioassay. That tightens the feedback loop between design and data, a loop that's usually painfully slow. When in a project does this help? Traditional wisdom says you save daring structural changes for the end, when you know the scaffold works. But by then, budgets are tight and timelines are rigid. Because this method is fast and cheap, teams can ask bolder what-if questions early, when the stakes are lower and creativity is higher. Current real-world plays. This isn't just a parlor trick anymore. One team built a fan of close cousins around an antibiotic lead, each with a small carbon handle ready for fluorescent tags or solubility boosters. Another group is poking stubborn peptides, editing a single position every other method bounced off. Analytical labs are eyeing trace-level tweaks during stability studies. Process chemists even imagine modular editing stations on pilot lines, diversify on demand instead of locking into one structure for the whole campaign. Long-term ripples. Zoom out and the possibilities get wild. Hospitals might tailor-fit drugs like a tailor hems a suit, trim or extend a chemical cuff for patients who metabolize quickly. Regulators could stress test safety by synthesizing plausible metabolites overnight. And the cultural shift? Chem students might start treating weird side reactions as treasure maps, not trash. Your mini assignment. Before next week, try this 3-part thought exercise. 1. If you had this radical late-stage editing tool, which one or two drug molecules would you tune first, and would you aim for solubility, selectivity, or metabolic stability? 2. Look at a reaction you're stuck on. Where could you deliberately relax the rules, change solvent, swap reagent order, play with light or temperature, to invite a productive mistake? 3. In your workflow this week, commit to testing one weird substitution you'd normally dismiss. Decide now what surprisingly good would look like, so you'll spot it when it appears. Let that simmer for a minute. So, here's what we covered today. First, a late-night lab slip in Cambridge turned basic blue light into a scalpel for drug molecules. Second, the reaction passes the real-world test. Crushed pills, syrups, deadline pressure, you name it. Third, it slashes time and cost on SAR exploration. Tweaking a chain is now as simple as changing a vial label. Fourth, teams across antibiotics, peptides, analytics, and process chemistry are already plugging it into everyday workflows. And finally, the mindset shift. Treat odd results as invitations, not inconveniences. Bottom line, you now have permission, and maybe a little challenge, to invite controlled chaos into your chemistry. Because the next blockbuster reaction might be hiding in the wrong experiment you're about to run. Take that curiosity back to the bench, and I'll meet you here next week.